We just learned all about light microscopy, which was the standard until the middle of the 20th century and remains tremendously useful today. But as we began to learn more about the microscopic world, using light microscopes that could visualize individual cells, we started to become curious about structures that are smaller than a cell, some details of which were difficult to visualize and study under standard light microscopes. Fortunately, electron microscopy, developed in the 1930s and 40s, proved to be much more powerful and allowed scientists to view these smaller structures. Some electron microscopy techniques employ a resolution of 0.2 nanometers, which is more than 1,000 times better than light microscopy. With this higher resolution, electron microscopes are able to magnify their specimen up to 1 million times their original size, allowing researchers to study proteins inside of cells and the molecular structure of viruses, all of which led to a better understanding of the microscopic world. Of course, we have to ask, how do they do this? Well, the answer is in the name. They use beams of electrons, which interact with the sample as the beam is transmitted through the specimen. As we recall from our study of chemistry, electrons are stable subatomic particles that carry a negative charge. Electron beams have a much shorter wavelength than the visible light that light microscopes use, which we refer to as the de Broglie wavelength, as we learned in the Modern Physics series. This shorter wavelength allows for a higher level of detail and resolution in imaging. One type of electron microscopy is called transmission electron microscopy, or TEM. For TEM, specimens are fixed on a support grid and either embedded in resin or stained with a material like uranyl acetate or compounds of heavy metals like osmium tetroxide. Certain structures within the specimen will take up more of the stain than other parts of the specimen, and those sections are called electron-dense regions. When electrons pass through electron-dense regions, they lose more energy compared to when they pass through sparser regions. This phenomenon is used by TEM to visualize the specimen at high levels of resolution and magnification. For the stained specimen to be viewed by TEM, it must be dehydrated, embedded in plastic resin, and cut into ultra-thin slices with a glass or diamond knife. The sections must be ultra-thin so that the electrons can pass through them. When the specimen is secured in the specimen holder, an electron beam is generated and shot out of the electron gun. The electron beam is accelerated down the column of the microscope after passing through the anode. The electron beam then passes through the specimen, once again losing energy as electrons pass through electron-dense regions. The electrons then pass through the objective lens, magnifying the image, before the electrons pass through the projector, or ocular lens, which magnifies the image further and projects it onto the fluorescent screen. This is where we start translating the electrons into an image. The fluorescent screen is coated with a chemical which appears as bright spots when electrons come into contact with it. The intensity of the bright spot depends on how much energy the electrons have when they hit the fluorescent screen. The camera under the fluorescent screen then captures the image made from the bright spots, which is displayed on a computer screen. Transmission electron microscopes are used to visualize structures that are too small to study with light microscopes, such as the cell organelles, proteins, cytoskeletal filaments, and the components of cell membranes. But even with the unmatched resolution and magnification abilities of TEM, it still does have its limitations. Some of these include the fact that you cannot study living specimens or image 3D structures, because the specimens must be dehydrated and cut into ultra-thin slices before imaging. The second type of electron microscopy is scanning electron microscopy, or SEM. With SEM, specimens are coated with vaporized gold or palladium ions, because when electrons come into contact with this coating, they cause atoms on the surface of the specimen to emit electrons. These emitted electrons are called secondary electrons, and these are what SEMs use to visualize their specimen. After the coated specimen is secured on the stage of the SEM, the process starts out similarly to TEM in that an electron beam is shot out and accelerated towards the specimen. In the SEM, the electron beam then sweeps or scans the surface of the specimen, which causes it to discharge secondary electrons. There are two types of detectors in the sample chamber: the secondary electron detector and the backscatter electron detector. The backscatter electron detector picks up backscatter electrons that come from deeper regions of the sample that are a result of elastic interactions between the beam and the sample. The results from this detector are primarily used to look at crystallography and the magnetic field of the sample, but for the purposes of microbiology, researchers focus on the detection of secondary electrons, which allow them to visualize the topography of the sample, or the distribution of parts and features on the surface of the sample. The detectors convert the electron signal into a light signal that is amplified by passing through a photomultiplier before the final image is projected on a computer screen. SEM imaging is primarily used to study the topography of cells and other similar structures. With this type of microscopy, researchers are able to investigate the depth of structures, which isn't possible with TEM, as SEM produces a 3D image opposed to the flat image from TEM. Yet, just like TEM, SEM cannot visualize live specimens, and SEM has worse resolution than TEM. Both TEM and SEM are expensive and require a lot of training, so they are not likely to be used apart from those pursuing a microbiology career. We covered quite a lot in this tutorial, so let's briefly summarize. Electron microscopy has around 1,000 times better resolution than light microscopy, because electron microscopy uses electron beams instead of light, which have a shorter wavelength. This allows some electron microscopes to visualize specimens at 1 million times their original size. Transmission electron microscopy, or TEM, records the differing energies that electrons possess after they pass through a specimen stained with heavy metals to visualize small, detailed structures, like proteins inside of cells, and molecules associated with viral particles. Scanning electron microscopy, or SEM, captures the secondary electrons that are emitted from the surface of samples coated in vaporized gold or palladium ions to visualize the topography of the specimen. And the main drawback, as we mentioned, is that both SEM and TEM cannot be used to visualize living specimens, and they are also quite expensive and complicated techniques to use. And with that, we know a bit about electron microscopy. Thanks for watching, guys. Subscribe to my channel for more tutorials. Support me on Patreon so I can keep making content. And as always, feel free to email me, ProfessorDaveExplains@gmail.com.